Okay, good morning Year 12. I hope you've had a good Easter break. You managed to have a bit of a relax, some time off with your family. It hasn't been too stressful for you. Welcome back to our virtual learning lessons. This week we're going to be focusing on the specific immune system and the immune response to the disease. I'm going to record a couple of different videos for you this week. The first of these is around antibodies and how antibodies work and their structure and how they defend the body. The second one will be around cell and humoral immunity, the immune response, and B and T lymphocytes. Okay, let's kick off with antibodies then. So, first thing we need to know is kind of the structure of the antibody itself. Okay, it's a fairly straightforward structure. But one you need to be able to fully understand. So I'm just going to draw that for you now. And this is something you're going to need to be able to recognise. Okay. Now you may recall some stuff about antibodies from year 11. One of those things being the kind of the general shape of the antibody, which is a kind of a regular Y-shaped formation. Just going to add some colour in here. Now antibodies consist of two what we call heavy chains and two light chains. So the light chains are a lot shorter than the heavy chains. I'll talk about those in a second. I'm just going to do the drawing first of all for you. Okay. Now it's important, you'll notice that I've marked out a kind of a break halfway along each of those chains. That's important. Okay, so a couple of features that we want to focus on first of all. Okay, so these are what we call our heavy chains. Okay, simply because they are larger. Okay, they consist of more amino acids, more bases, and therefore we class them as heavy chains. And as you can imagine, these pink ones are the light chains. Okay. Now, there are certain other regions that we need to focus on. Okay, the first is this little part here. So, where you notice that I've drawn and left blank. Okay, this part here, which is the same on both of the chains. This is what we call the variable region. Okay, variable region. Now, the reason it's called that is because it is different in every antibody. Okay, so every type of antibody, every different type of antibody that you have in your body has a different variable region. This section at the end there, this tiny little section. Okay, consequently, this part here, which is the shaded part, okay, is what we call the constant region. Okay, and this part is the same. Okay, so this is the same in all antibodies. So you can see the majority of the antibody is very similar. Okay, no matter which type of antibody we have, and there are millions of different types of antibodies in your body, they are mostly similar. It's just these parts at the end that are different. And these parts at the end are the critical parts, because they host this little bit here, which is what we call the antigen binding site. Okay, antigen binding site, and this is the critical part, because this is where the antibody will lock on to the invading antigen, or the invading pathogen and its antigen, and be able to carry out its mode of action. Okay, so these variable regions, these light and heavy chains, are polypeptide chains. Okay, and they are held together in between and within, okay, by these little disulfide bridges, which we have heard about all before. Okay, we have an understanding of disulfide bridges from know the structure of DNA. Okay. Now then, some critical features, okay, of antibodies. Number one, they are glycoproteins. Can't spell, sorry. Glycoproteins. And what we call immunoglobulins, okay, or IgG. Okay, this is a critical part. There are lots of different antibodies, some called IgM, IgA, IgG, IgE, but the IgGs are the ones we're really concentrating on at the moment. Okay, so what do? They bind to specific antigens on the invading pathogen or its toxin. Okay. As I mentioned, there is a specific antibody for each individual antigen, okay, that invades your body. So theoretically, no matter what you come into contact with, there will be an antibody to fight that, unless of course your body has never come into contact with that before, and that is obviously the problem that is ongoing at the moment with COVID-19, is that this is a brand new pathogen, a brand new antigen, and so people's bodies don't have the antibodies in them already to be able to fight that. Okay. Now the way they work, very simply, and we'll go into a little bit more detail in a moment, is that they bind, okay, with similar to enzymes, with the kind of a lock and key mechanism. Okay. So it's a similar process to enzyme substrate. And in a similar way, they therefore forms an antigen antibody complex. Okay. So some key features. Now, a couple of other things. How do they actually work? So, a few key points. Okay. First of all, is that the antigen antibody complex acts as something called an opsonin. Okay. What this means is it makes the complex easy to be engulfed. Now, if you remember from when we were, I was talking about phagocytosis and the process of phagocytosis, where phagocyte is made. This is the thing which goes around and collects up all of the toxins and the pathogens in non-specific immunity, and those phagocytes are still there, and they are looking out for all of this, these complexes. Okay, but they're only really able to be collected and found if when this antibody antigen complex acts as this opsonin molecule. It means that therefore the complex can no longer invade host cells. Okay, so it renders it kind of ineffective. Okay. So I should say pathogens when part of complex. So most pathogens can no longer invade host cells when they're part of the complex. They also act as something called an agglutinin. Okay, and what that does is it causes the complexes to clump together. It just makes it easier for the phagocytes to work. Okay, and then finally, okay, they can also act as an antitoxin. So we remember that all pathogens are able to, the way they work is through a number of different methods, and one of those is to produce toxins, okay, which then go and invade host cells. So these antibodies can act as antitoxins, which bind to the toxins produced by pathogens. Okay, so that's the basis of antibodies and how antibodies work. Now, for these antibodies to actually take hold, takes a bit of time. It is a slower process than the non-specific response that we talked about previously. Okay, it can take up to maybe a couple of weeks for this to actually happen. Okay, so from first being attacked by the virus, it can take up to a couple of weeks for these antibodies to really kick in and fight the virus. Which is why, when, for example, you meet something that you've already had before, so for instance the common cold, okay, you're still going to get the cold, but you are because it takes a little while for your antibodies to kick in and to actually work. Okay, but if it's the same pathogen straight away, okay, this happens a lot quicker. Once those antibodies are there, you have something called immune memory cells, which fight a lot quicker. And we're going to come on to that in the second video and talk about lymphocytes and how white blood cells are able to fight disease a little bit more detail. Okay, that's it for this video on antibodies, and I will, as I say, the next one we'll talk about the more wider specific immune response.